I'd like to thank my hosts for inviting me to make this presentation about one of the great scientists of Romanian history, George Palade. But I'd like to start first by congratulating the Moldovan Academy on the 60th anniversary of your formation. That's a tribute that you've been able to carry on for so long under changes of leadership. I'd also like to thank the Academy for honoring me by making me an honorary member. Of course, because of travel restrictions, it's not been possible for me to come, but I promise that next summer I will resurrect the trip that I had planned for last summer, and I'll look forward to meeting you all in person after having met some of you at least virtually. Well, let me tell you a bit about George Palade and the history of his scientific achievements and my own relationship with him. I've known Dr. Palade since I was a graduate student in the early 1970s. I met him because he was a very close friend and colleague of my graduate mentor, Arthur Kornberg. Kornberg, the year before I started graduate school in 1970, he did a sabbatical, a short sabbatical period of study in Dr. Palade's group, working with one of Palade's proteges, David Sabatini, about whom I'll speak later. And so Palade then returned the favor by coming to Stanford, where I was a graduate student, and giving several lectures where I met him. But as you'll see, I only became very directly professionally interested in his work a few years later. So let me begin by telling you a bit about what Dr. Palade did. Of course, he trained in Romania through a medical degree in Bucharest, but he immigrated to the US in the 1950s, where he established his research program based on the use of this instrument that he's seated behind. It's called an electron microscope. It was invented in Germany in the early part of the 20th century, but it was applied to biology only through the talents and perseverance of Palade and his proteges. Palade, in the mid-1950s, used this instrument to be the first to visualize ribosomes inside of a cell that had been fixed and prepared as a specimen for electron microscopy. It was only subsequently demonstrated by Palade and others that these ribosomes, these granular particles in the cytoplasm of cells, are responsible for the biosynthesis of proteins. Palade then did much of his really most pioneering work as a professor in the Rockefeller University, also in New York, where he was living, where he, in a series of really technically challenging but beautiful experiments that I'll summarize for you in the next few slides, was able to demonstrate in the pancreas, largely in experimental animals, that proteins that are secreted, molecules like insulin that I'll allude to, but other proteins that are secreted into the gut that aid in digestion, are made through an elaborate network of membranes that connect to each other, it turns out, in all eukaryotic cells. It's called the secretory pathway. And the crowning recognition of his achievements, together with Albert Claude and Christian Dedeuve, was the award of the Nobel Prize in Physiology or Medicine in 1974, the year I finished graduate school. The paper that he published the following year in Science magazine summarized all of his work, really. And it was in my lab, as I was beginning my own career at around that time, this paper of Palade's became the Bible. I assigned it to all of my students to read and to commit to memory, because it really describes in broad terms the brilliant experiments that he and his colleagues did to visualize molecules like chymotrypsin, chymotrypsinogen, proteins, proteases that are made in the secretory pathway and pass from station to station within the cell. Let me summarize some of the experiments that led to his observation. First, through a laborious series of techniques that were perfected by his protege, David Sabatini, it became possible to preserve the intracellular membranes in tissues of the pancreas and to observe, as you see here, the elaborate network of membranes that constitute the structure called the endoplasmic reticulum. This structure had, of course, been visualized previously by light microscopy, but the resolution possible with electron microscopy allowed the refinement of our understanding of this organelle. And here you see a high resolution image of a network of ER membranes with ribosomes bound on the cytoplasmic surface of the membrane. Here in even higher resolution, you see these ribosomes. Now, Palade and some other experiments using visualization and cell fractionation was able to show that newly synthesized proteins that were eventually going to be secreted from the cell were made by ribosomes bound on the cytoplasmic surface of the endoplasmic reticulum, where the polypeptide would then be moved vectorially into the interior, the clear lumen of this organelle, fold and become glycosylated and form disulfide bonds, as he predicted here. Further, he speculated quite presciently that there must exist in this membrane a channel through which these polypeptides could traverse, thus avoiding the hydrophobic interior of the membrane. For soluble secreted proteins, they must somehow pass through the bilayer, but that could not happen were it not for a channel. It was left to his younger protege, Günter Blobel, to really demonstrate this experimentally in work that won him a Nobel Prize some years later. Now, another really beautiful and really very challenging series of experiments were conducted by other proteges of Palade's, Jim Jameson and Lucien Caro. They were able to show that in tissues, pancreatic tissue slices, they could add C14 labeled amino acids in a short period of incubation where these tissues and cells remain viable. And the samples were then fixed, chemically fixed after just a 1 or 2 minute brief period of radiolabeling. The samples were then, the fixed and processed samples were then put onto an ultra thin photographic emulsion that Lucien Caro had perfected and left in the dark for months to allow tracks of C14 radioactive decay to register by a silver grain deposit on the thin photographic emulsion. They could then take the sample afterwards and superimpose the photographic emulsion with a few grain tracks on top of the thin section of the cells and show that the initial pulse of radioactivity populated this structure that was rich in the membrane endoplasmic reticulum. If the initial pulse sample was then allowed to incubate in the presence of a great excess of non-radioactive amino acids, they could observe where these radioactive proteins migrated in a kind of a pulse chase regimen by taking samples at different times, fixing those samples, and then processing them in the same way. And you see in the image on the right, after about 30 minutes, the grain tracks that initially appeared in the endoplasmic reticulum region of this cell called the pancreatic Eisner cell now appear in immature granules, secretory granules that will eventually mature into granules that can allow these proteins to be discharged. So there was some movement of proteins within the cell. Now, closer inspection showed this progression of the movement of grain tracks from an initial disposition around the rough endoplasmic reticulum to a region surrounding another membrane called the Golgi apparatus, which had been observed decades earlier by light microscopy, but whose function was completely unknown until Palade demonstrated that it was a temporal stage in the movement of secreted proteins. And finally, after some minutes more, the radioactivity appeared over these immature granules, and ultimately after a couple of hours in mature granules where they were then, these proteins were then housed and stored for stimulated discharge. Now this same process is used by beta cells of the pancreas to manufacture and secrete insulin. And here you see, this is not a micrograph of Palade, but of another really brilliant morphologist by the name of Lele Orci, an Italian scientist. This is a section of the beta cell of the pancreas showing the mature granular form of insulin housed within these granules, all surrounded by a biological membrane. And when zymogen contents are eventually secreted, Palade showed, and others subsequently showed, that the granular material housed within a membrane comes to the surface, the cytoplasmic surface of the membrane that surrounds the cell, called the plasma membrane. And at a certain point, these membranes merge by a process called fusion. And in this case, the insulin granule is discharged to the cell exterior. Now, in this 1975 paper of Palade's, he was able to demonstrate this progression of movement through the secretory pathway, and even to make some speculations about the machinery that may be necessary to pinch a vesicle off of a membrane as an intermediate to deliver it to this station, the Golgi apparatus, or to a granule that may be condensing this content. So this was all very prescient. And I remember quite vividly in 1974, in the week after Dr. Palade received his Nobel Prize in Stockholm, he traveled to San Diego in California to deliver a very special lecture to the society that I was just becoming a member of, called the American Society for Cell Biology. And so he delivered the lecture, including the images that I've shown you, to a very appreciative audience. And in the last paragraph of this seminal review, he speculates what will transpire next in the developing field of membrane traffic. He speculated that there may be another role for the secretory process in a general but still unrecognized function. And let me read, he said, among the non-secretory functions postulated for the endoplasmic reticulum and the Golgi complex is the production of cellular membranes, including the plasma membrane. And this is only a suggestion, and it was based purely on morphologic evidence. However, it will become necessary, he said, to develop more comprehensive data on the chemistry and function of different membranes in the secretory pathway. Now, I remember his lecture vividly. It was quite a treat because he is, as many of you may have heard, a very elegant and deeply scholarly person. He was so greatly appreciated by the audience at the meeting that he received a standing ovation at the end of his lecture. He was a very modest man, but clearly this was an appreciation of all that he had done to build cell biology into the modern era. Now, I had just come from my graduate work with Arthur Kornberg, where I had really focused entirely on a biochemical approach, where one takes a complex pathway apart into its molecular constituents. And what I saw at the end of Palade's lecture was an opportunity, because as he said in this concluding paragraph, it will be necessary to have a comprehensive data on the chemistry and function of different membranes. And in 1974, at the height of his powers, in spite of the beauty of this work, we knew nothing about the mechanism, the molecular mechanisms involved in the movement of a protein like insulin from its site of synthesis outside of the cell. So I began my career at UC Berkeley in the fall of 1976. And just 2 years later, Palade, who was greatly appreciated by my colleagues here, invited him to give an honorific lecture. This is in 1978, in May of 1978, where he talked about his work in characterizing the secretory pathway. And I had the great opportunity to meet him on that occasion as a professional colleague. Before that, I had been a student. And I had the thrill of telling him about our work on protein secretion in yeast. He was not aware that yeast cells secrete glycoproteins. And so it was a treat to be able to tell him how we were going to approach the problem. That was a very busy time for Dr. Palade, having just won his Nobel Prize. He made many trips around the world, and one that you may have heard of was on the occasion in 1979 when he returned to Bucharest to celebrate the opening of a new Institute of Cellular and Cell Biology and Pathology, together with his wife, Marilyn Farquhar, his proteges, Gunter Blobel and David Sabatini, his fellow laureate, Christian Deduve, and most importantly for you today, his close colleagues from Romania, Nicolae and Maya Simonescu. They spent many years collaborating with Dr. Palade in his lab in New York, but then they were the founding organizers of this very important institute in Bucharest that I believe continues to this day. So this is an amazing picture of history. Unfortunately, many of the individuals in the front row are no longer with us. David Sabatini, on the other hand, just recently celebrated his 90th birthday in New York. Well, I'm going to just briefly tell you about the connection that Palade made to me for my work. When I began at Berkeley in 1976, I was struck, of course, by the power of what Palade had done, but the promise of understanding this process at a molecular mechanistic level, I felt required using a different source material, not just animal pancreas, but a simpler organism, Baker's yeast. And Baker's yeast is a simple genetic system. It's possible to do both genetics and biochemistry. And it was already fairly well appreciated at that point that yeast had an evolutionary connection to man. There was a fundamental conservation in processes, particularly those within a cell, that would be shared between a yeast cell and a human cell. And so I began my career by looking at yeast cells to see how they may secrete proteins, obviously not secreting insulin, but secreting proteins that they need for their own growth. Now, we knew at the time that it was possible to study essential genes because those that were involved in the logic and progression of cell division had already been obtained. And it was really easy to study yeast, very inexpensive. You can grow very large quantities. There's a very regular cell cycle. A daughter cell forms on the surface of a bud and grows in preference to the mother cell over the 90 minutes or so that it takes a cell to divide, and then they divide in half. If you look inside of a yeast cell, it looks very different than a pancreatic exocrine or beta cell. The cytoplasm is filled with ribosomes. There are some organelles that are characteristic of eukaryotes, including the endoplasmic reticulum membrane. But Peter Novick, one of my first graduate students, and I were particularly interested in this collection of small vesicles that congregate under the bud portion of a dividing cell. We speculated that these vesicles would carry secreted enzymes into the wall that surrounds the yeast cell that would allow it to scavenge carbohydrates from the environment. But further, we speculated that these vesicles with their membrane surrounding could be the building block of the cell surface. And recall, Palade had speculated with no evidence that secretory vesicles may be responsible for the assembly of the plasma membrane. But I felt that with yeast, we had an opportunity to test that hypothesis by looking for mutations that block the production or movement or fusion of these vesicles at the cell surface. If those mutations block secretion, they may also block plasma membrane assembly. And just very briefly, we were able to do this. We were able to isolate such mutants. And we had our first mutant called SEC1, short for secretion 1, which blocked secretion and blocked and killed cells. And just at that time, Palade was visiting Berkeley to give the lectures that I mentioned a few minutes ago. And my student, Peter Novick, visited with Dr. Palade, telling him about his work. And Palade said, well, now, of course, you must examine the mutant as it is blocked in secretion to see what the cell looks like inside. And one of the great thrills of my career came several months later when Peter Novick called me down to the electron microscope room in the basement of our building to show me this cell. This is the SEC1 mutant cell. It's dying, secretion is blocked, and what happens is that secretory vesicles now build up, filling up the entire cytoplasmic volume. No longer just a few under the bud portion. The cell becomes chock full of these vesicles, and eventually it chokes because it can no longer grow and divide. Now, we then repeated our genetic approach in many other studies that I don't have time to tell you about to demonstrate that there is a pathway very similar to that Palade had described in mammalian cells that allows yeast cells to manufacture, transport, and then secrete secretory proteins into this part of the cell. Of course, what we were able to show is that each step is populated by a number of genes required for each of the steps in this pathway. This was not possible to do in a mammalian cell, but it was very possible to do in yeast. Further, we were able to show by molecular cloning of these genes that they were virtually the same as corresponding genes that had been identified subsequently in the sequencing of the human genome. And therefore, it became clear, as I had mentioned a few moments ago, that this pathway was fundamentally conserved over the 2 billion years or so of evolution. So much so that it became possible to use yeast as a platform for the production and secretion of human proteins that are recognized by the pathway and secreted. And now, many years later, it's possible to manufacture human insulin by a fermentation process. Here's an example where yeast cells grown in very large quantities are grown on industrial scale to produce now 1/3 of the world's supply of human recombinant insulin and many other useful proteins. The hepatitis B vaccine that is so important is produced entirely by its synthesis in yeast and integration into these little vesicles that yeast cells make. Well, I'm going to short circuit all of that and go back to Palade because he really is the reason for this lecture and the reason that I have made the progress that I have. Palade, of course, continued in his career. He moved to the University of California, San Diego, where I met him on several occasions, and I'll conclude with that in a few minutes. But I had the really rare opportunity some years after my work had grown to deliver the George Palade lecture at Yale University. Yale was the place that Palade moved for some years after he left Rockefeller University. He started a new department of cell biology and trained another generation of great cell biologists. In fact, at that point, my former student, Peter Novick, the one who had isolated all of the mutants that got my career going, he had moved with his wife, Susan Farrow Novick, to take positions as professors at Yale. And Peter was my host. And I have this memory of that occasion now 13 years ago, where I was asked to deliver the Palade lecture. Well, later that year in 2008, Dr. Palade died after some years of failing health. He was still at the University of California, San Diego. And I was asked and quite honored to write an essay about his life and his career as a perspective in Science magazine, which was published just in the weeks after he died. And you can look at this if you wish, but I want to highlight two things about this essay that I wrote because it reflects the deep scholarship and wit of Dr. Palade. I had many occasions to share the podium with Palade at various symposia, and he was always remarkably gifted in his eloquence. On this occasion that I will refer to, I had once seen him deliver a lecture at a meeting in Strasbourg in 1983 And the tradition in France at all scientific meetings back then was that the concluding lecture had to be given in French. All the rest of the meeting was in English. And so they turned to Palade to give the concluding lecture, which he delivered in eloquent French, but he could have delivered the lecture in many other languages. And in fact, I bet he could have delivered it in Latin. In fact, I knew that Palade, one of his hobbies was to study ancient Roman military history. Some years later, at a major symposium at Cold Spring Harbor on protein traffic, Palade was again asked to give the final lecture. And in his written statement at that meeting, after that meeting, he made he revealed his deep history with a very witty comment that I'd like to share with you. So in 1995, this Cold Spring Harbor Symposium, where he had reached the age of 83, he mused that according to the 17th century French literature, El Cid Campeador, hero of the Spanish wars against the Moors, returned to court after a fierce and long battle. Quote, according to Palade, long, but not as long as this symposium, as Palade put it, to tell his king that the battle ended because there were no fighters left. Palade then goes on to say, as the days of this symposium wore on, Palade hoped that by the time he was scheduled to speak at the end of the conference would be finished because there would no longer be any combatants, any speakers left at the symposium, and therefore his presentation would not be called for. Fortunately, that was not true, and he delivered a very comprehensive picture of the whole field as of 1995. Well, finally, the last time I saw George was at a symposium in his honor at the age of 90, held at the University of California, San Diego. And here we are speaking. As I said, for the last years of his life, very unfortunately, Palade was in slow declining health. He eventually had Parkinson's disease. He lost his vision and he became immobile. But he was still there at age 90, still very interested in what was going on. Also, his wife, Marilyn Farquhar, was at this symposium. Regrettably, she died just a couple of years ago. And finally, we're left with a great man himself at age 90, clearly having suffered from the aging process, but still sharp and ever the man that we have known to grow in love. He was a remarkable man. He brought great statesmanship and precision and intuition to a newly emerging area of cell biology. He is really the father of modern cell biology, and I salute him and the memory that he brings to this day. So thank you all for your attention.